What is gene silencing and what is the importance of that? We all develop from a single cell and that is called zygote. Zygote contains about 50,000 genes, all of which are active in zygote. Now from that one cell, two cells are generated by the process of cell division. From those two cells, again 4 cells are generated, then 16 cells, then 32 cells, and likewise all the trillions of our body cells are generated. As we develop in mother's womb, some of those 50,000 genes become silenced in some body parts. Let me present a very simple example. Genophyte, that is responsible for the height of a human being. Now that gene is not required for the development of liver inside our body. Therefore, when liver cells are generated in our body or during the development of the embryo in mother's womb, the gene of height will become silenced. Another example is the gene for skin color. Again, that gene is not needed for the liver cells because the skin color of liver has no importance at all. That's why in liver cells, the gene of skin color is also silenced. On the other hand, the liver cells can generate some enzymes which help to digest food in us. The genes for those enzymes are not required to be expressed in our skin. So in our skin cells, those genes for the enzymes remain silenced. So what is the mechanism for gene silencing? Obviously, the first and foremost one is gene mutation. Mutation is a very sudden change in the genes. Our genes are nothing but short parts of DNA, and DNA is made up of a few chemicals: adenine, guanine, thymine, cytosine. These nitrogenous bases are used for making the gene sequence, and obviously there are sugar and phosphate. Adenine, guanine, thymine, cytosine or AGTC, these things can make a specific sequence which is called a gene and which can generate some proteins, etc., which are responsible for the characters seen in us. Now mutation is a sudden change in that sequence of AGTC or ATGC, whatever it is. If that type of change occurs and the protein is not produced at all, or a defective protein is produced from that sequence, then the required character or the desired character is not seen in us. So that is a kind of gene silencing where the desired protein is not produced and the process is mutation, the change of sequence of adenine, guanine, thymine or cytosine in the gene itself. Now the second mechanism is called epigenetic mechanism. Epi means outside, epigenetic, so outside the gene, the genetic sequence of ATGC, AGTC, etc., that is not changed, but something occurs from outside and that will prevent the expression of the gene and the required protein, the desired protein is not produced. So the gene becomes silenced. The most well-studied and well-known example of epigenetic modification is DNA methylation. DNA strands have a polarity from 5' to 3'. Now, if a cytosine or C is present in the 5' side and a guanine or G is present just after that, so the sequence is 5' C, G, 3'. In that kind of regions, the 5th carbon of cytosine can have a methyl group instead of a simple hydrogen. That is called a methylated cytosine or 5-methyl cytosine. That creates the DNA methylation and this will help to silence the gene. Here I must say another thing, that is DNA methylation is very much related to histone modifications. Histones are some positively charged proteins which are present along with DNA and can help the DNA to be packed inside the nucleus. In one of my later videos, I will discuss the histone modification process in a greater detail. In this video, just I want to remember that at least 4 different types of histone modifications are identified by scientists, and all these are related to DNA methylation. Now let me come to the next type of gene silencing mechanism, and that is also called as post-transcriptional modification or post-transcriptional gene silencing. Transcription is the process by which genes can produce messenger RNAs or mRNAs, and these mRNAs are used to code for the protein which helps us to produce the character in us. Now suppose no mutation has occurred, no gene silencing by epigenetic mechanisms has occurred. The mRNA is generated from the gene. Now this mRNA can again be silenced by a process which is called post-transcriptional gene silencing, and that is mediated by some very tiny RNAs which are called microRNAs or miRNAs. MicroRNAs can target the specific regions of mRNAs and can destroy those. So the mRNAs which are generated from the intact gene will not produce the desired proteins, and therefore the gene will be called as silenced. So I hope that now you have understood the different types of gene silencing mechanisms which can occur in human beings. That's all for this video. If you have any question, write those in comments. If you like my video, please subscribe my channel, press the like button and share my video. So till my next video, stay safe, stay tuned and bye.